Hello viewers, hope you are safe and utilizing quarantine times to upgrade yourself. So this video is about glycogen metabolism pathway, and in this video we are going to learn about a short introduction about glycogen, glycogenesis and glycogenolysis. So before moving forward, we will just see about the topic glycogen. So what basically is a glycogen? Glycogen is a reservoir of carbohydrate in animal. That's why it is often referred as animal starch. It is present in liver in a very high concentration and then in a muscle and in a brain. It is required to meet the immediate energy demands of the body. Glucose is the repeating unit in glycogen joined together by alpha 1,4 glycosidic bonds and alpha 1,6 glycosidic bonds at branching points. As you see the structure of glycogen on the top of the screen on the right hand side is there. But if you want to see the structure of glycogen for better understanding of alpha 1,4 glycosidic bond and alpha 1,6 glycosidic bonds, it is in the screen right now. In this, there is a linear chain of glucose having 1,4 glycosidic linkages and the branch chain are having 1,4 glycosidic linkage. The alpha 1,6 glycosidic linkage are only present in the branching points. Rest of the bonds are alpha 1,4 glycosidic bonds. So now we'll move on to the next topic, that is glycogenesis. So the synthesis of glycogen from glucose is known as glycogenesis. Glycogenesis can take place in cytosol and requires ATP and UTP besides glucose. So when you see glycogenesis basically occurs in 4 steps, that are synthesis of UTP glucose, requirement of primer to initiate glycogenesis, number 3 is glycogen synthesis by glycogen synthase, and the number 4 is formation of branches in glycogen. So now we will discuss the steps elaborately. At the first is synthesis of UTP glucose. In this, there is a pathway where glucose forms intermediates before forming UTPG. The first reaction is glucose is being converted to glucose 6-phosphate by the help of glucokinase or hexokinase. The enzyme hexokinase is present in the muscle, but the glucokinase is present in the liver. And the second reaction is the glucose 6-phosphate is converted to glucose 1-phosphate by phosphoglucomutase. And in the third reaction, the glucose 1-phosphate is converted to uridine diphosphate glucose, that is UDPG, in the presence of UTP by the enzyme UDP glucopyrophosphopyrolase. Clear? It was about the first step. In this, the glucose was converted to uridine diphosphate glucose. In short, it is known as UDPG. Now, the second step is the requirement of primer to initiate glycogenesis. A small fragment of pre-existing glycogen must act as a primer to initiate glycogen synthesis. But in the absence of glycogen primer, a specific protein named glycogenin have the ability to accept glucose from UDPG. In the glycogenin, the amino acid tyrosine is present, which is having hydroxyl group. And to that hydroxyl group, the glucose unit can be attached from UDPG. The enzyme named glycogen initiator synthase transfers the first molecule of glucose to glycogenin. Then after that, glycogenin itself can take up a few glucose residue to form a fragment of primer which can serve as an acceptor for the rest of the glucose molecule. So in this step, you see the primer was there, the fragment of glycogen acts as a primer, but if there is no primer present or the fragment of glycogen is not there, then with the help of glycogenin we were able to make a primer with the help of glycogen initiator synthase. Now there is other step, that is third one. Now the third step is glycogen synthesis by glycogen synthase. In this, the enzyme glycogen synthase is the key enzyme that is responsible for the formation of 1,4-glycosidic linkages. This enzyme will transfer the glucose from UDP glucose to the non-reducing end of glycogen to form a alpha 1,4 linkages. So in this step, with the help of glycogen synthesis, simply the formation of alpha 1,4 glycosidic bond was done in the glycogenin. Now, the last step of glycogenesis is the formation of branches in glycogen. Glycogen synthesis is only able to form a linear unbranched molecule with alpha 1,4 glycosidic linkages. However, the glycogen is having a branched tree-like structure. Therefore, this enzyme is not sufficient to make the branching. The formation of branches is brought about by the action of branching enzymes, namely glucosyl alpha 4,6 transferase. This enzyme transfers a small fragment of 5 to 8 glucose residue from the non-reducing end of glycogen chain by breaking alpha 1,4 linkages and by joining that residue with another glucose residue where it is linked by alpha 1,6 bond. This leads to a formation of new non-reducing end besides the existing one, and the bond formed are alpha 1,6 bond. After that, glycogen is further elongated and branched respectively by the enzyme glycogen synthesis and glycosyl 4,6 transferase. So by this, the glycogenesis is over, but now we will see the pathway of glycogenesis to make it more clear to you guys. When you see the pathway, the glucose is converted to glucose 6-phosphate with the help of glucokinase, and then the glucose 6-phosphate is converted to glucose 1-phosphate and then subsequently to UDP glucose. Once the UDP glucose is ready, it will now donate that glucose for the formation of glycogen. So if there is no primer, by the help of glycogen and the glucose of the UDP and in the presence of glycogen initiate a synthesis, the glycogen primer is ready. After the glycogen primer is ready, with the help of glycogen synthesis over here, we can see that it can form 1,4 glycosidic bonds. It can form 1,4 glycosidic bond, and it can form a straight linear chain of a initial portion of a glycogen. But it is having a branch structure, so therefore we need some branches. So for the branching, we use the other enzyme, that is glucosyl alpha 4,6 transferase. With the help of glucosyl alpha 4,6 transferase, it cleaves 1,4 bond having 5 to 6 or 5 to 8 residues of glucose, and then that residue is attached somewhere in the glucose by forming 1,6 glycosidic bond. And then with the help of that two enzymes, this glycogen synthesis and glycosyl transferase, this elongation and branching takes place until the glycogen structure is formed. Now our next topic is glycogenolysis. The degradation of stored glycogen to glucose in liver and muscle constitute glycogenolysis. In simple language, it is just said that the formation of glucose from glycogen or breakdown of glycogen is known as glycogenolysis. The pathway for the synthesis and degradation of glycogen are not reversible. And there are a set of enzymes that are independent which carry out glycogenolysis inside the cytosol. Glycogen is mainly degraded by alpha 1,4 and alpha 1,6 glycosidic bonds. After that, the glycogenesis, I mean to say, after that, the glycogenolysis takes place. Glycogenolysis basically occurs in 3 steps, that is shown in the video right now. That is the action of glycogen phosphorylase, action of debranching enzyme, and the formation of glucose 6-phosphate and glucose. Now we will see this steps and discuss it elaborately. The first step is the action of glycogen phosphorylase. Now in this step, the enzyme glycogen phosphorylase cleaves the alpha 1,4 glycosidic bond from the non-reducing end sequentially to yield glucose 1 phosphate. This process is called phosphorylosis and it continues until the limit dextrin is formed, which cannot be further degraded by phosphorylase. So what is limit dextrin? Limit dextrin is a type of a glycogen with four glucose residue on either side of branching point. Branching point means where there is a alpha 1,6 glycosidic link. So from that, in the both the direction or in the either side, there is four glucose residue. So that type of glycogen is known as limit dextrin. So in the first step, we saw that with the help of glycogen phosphorylase, the glycogen were broken down or cleaved sequentially until the limit dextrin was formed. Now we'll move towards the second step. The second step is the action of debranching enzyme. The branches of glycogen are cleaved by two enzyme activities called debranching enzyme, hence it is a bifunctional enzyme. The enzyme named glucosyl 4,4 transferase removes a fragment of 3 or 4 glucose residue attached to a branch and transfer them to the another chain. That is the linear chain. In this step, one bond of alpha 1,4 glycosidic linkage is cleaved and at the same time one bond of alpha 1,4 glycosidic linkage made, but at the different places. Now with the help of amylo alpha 1,6 glucosidase, it breaks the alpha 1,6 bond at the branch having a single glucose residue and releases a free glucose. This second step in which the amylo alpha 1,6 glucosidase is acting is only it only works when all the side chain are reduced. I mean to say there is no 1,4 glycosidic link. There is only one glucose that is linked with linked with the straight chain with 1,6 bond. So the main aim of this amylo alpha 1,6 glucosidase is to break that alpha 1,6 bond. Clear? Now once the glucosyl 4:4 transferase does its work for cleaving this 3 or 4 glucose residue from the branch chain to making it a linear chain, and after that amylo alpha 1,6 glucosidase breaks that one alpha 1,6 bond and releases a free glucose. After that, the remaining molecule of glycogen is again available for the action of phosphorylase and debranching enzyme. To repeat the reaction is stated in the step 1 and step 2. The step 1 is the action of glycogen phosphorylase and the number 2 is the action of debranching enzyme. Now, the last step is the formation of glucose 6-phosphate and glucose. Now, through the combined action of glycogen phosphorylase and debranching enzyme, glucose 1-phosphate and free glucose are produced in the ratio of 8:1. Glucose 1-phosphate is converted to glucose 6-phosphate by the enzyme phosphoglucomutase. After that, lastly, with the help of enzyme glucose 6-phosphatase, the glucose 6-phosphate is converted to glucose. But the fate of glucose 6-phosphate depends on the tissue. The liver, kidney, and intestine contains the enzyme glucose 6-phosphate that cleaves the glucose 6-phosphate to glucose. Whereas the enzyme is absent in muscle and brain, hence free glucose cannot be produced from glucose 6-phosphate in this tissue. Therefore, liver is the major glycogen storage organ to provide glucose in the circulation to be utilized by various tissues. Now, when you see the pathway of glycogenolysis, it might be clear to you guys. So let me explain all these 3 steps again by looking into this pathway. So the initially our first step was the action of glycogen phosphorylase. So it is the glycogen with the help of glycogen phosphorylase. The limit dextrin is produced. Limit dextrin is produced because it cleaves all the 1,4 glycosidic bond, and from that the branching only 4 glucose moieties are present, making it a limit dextrin. And after that, with the help of the debranching enzyme, the transferase enzyme, what it does is from the limit dextrin, the branching that is shown in the three dots over here, that three dots is a three molecule or a three residue of glucose. So that residue is cleaved at the second I mean to say the 1,6 bond is not cleaved, but 1,4 bond is cleaved over here. So from there, that three molecules of glucose is cleaved, and at the same time that three molecule comes and bind, forming a longer linear chain, and it was the activity of the debranching enzyme transferase activity. After that, there is the other one, that is amylo alpha 1,6 glucosidase. What it does is it cleaves the glucose that is present. It cleaves the glucose that is present, that is resembling with the blue dot, because it is only having 1,6 glycosidic linkage. So when this activity of this enzyme works, that is cleaved, and that free glucose is released by forming a straight chain. Again, by the further action of phosphorylase, we get this ratio of free glucose and glucose 1 phosphate at the ratio of 8:1, and then by the help of this phosphoglucomutase and glucose 6 phosphate, we get a glucose. Hope with this explanation, I think you guys have understood what is glycogenesis and what is glycogenolysis. So in a simple term, you can say that glycogenesis is a formation of glycogen from glucose, formation of glycogen from glucose, and in a glycogenolysis, lysis means breakdown. It is a breakdown of glycogen into glucose, or just in a simple manner, you can say that it is a degradation of glycogen. Clear? So this much for today. Thank you very much for watching this video, and hope to provide with some other materials in upcoming days. Thank you.